Our bodies run on energy. Even as you sit watching this, your body is generating enough energy to power 7 10-watt light bulbs. Most of that energy is provided by tiny structures called mitochondria present inside our cells. These mitochondria are the powerhouses of the human body. They take fat, sugar, and protein from our food and combine it with oxygen, converting it into energy for our cells and tissues, such as brain and muscle. Mitochondria have their own DNA that's crucial to this energy conversion process. This is different to the DNA found in the nucleus. While nuclear DNA determines our physical characteristics, mitochondrial DNA does not. But both types of DNA must be healthy for the mitochondria to function effectively. Faults in either can cause mitochondria to stop working properly, preventing them from converting fuel into energy. If the number of faulty mitochondria reaches a critical level, our cells begin to run out of energy, fail, and even die. Since mitochondria perform so many different functions, there are literally hundreds of different mitochondrial diseases. The effects include fatigue, speech disorders, hearing difficulties, muscle weakness, heart problems, liver disease, bowel problems, and sometimes, in very severe cases, it may even be fatal. The sheer variety of symptoms associated with mitochondrial disease makes it hard to diagnose. Despite this, we're making rapid advances every day in our understanding of how it develops and is passed on, all of which help us to devise new strategies to prevent and treat the disease in the future.
Professor Seyfried, okay, he's the author of this book right here, Cancer as a Metabolic Disease. This is my new favorite book, incredible. You need a little bit of biochemistry to get through it, but there's a lot of parts that are really easy to understand. And Professor Seyfried has taught and conducted research in the field of neurogenetics, neurochemistry, and cancer for more than 25 years at Yale University, Boston College. He's published more than 150 scientific articles and book chapters and is on the editorial boards of Nutrition and Metabolism, Journal of Lipid Research, Neurochemical Research, and ASN Neuro. So welcome, and I really appreciate you spending some time talking with me today.
Oh, thanks, Eric. It's nice to be here.
Absolutely. I think it's important to start out, if we could, on the big idea here, where, and kind of, if you could do it in layperson's language, just to describe the difference between a normal cell and cancer cell. I think that's important to just talk about that.
Yeah, well, there are many differences. The prime difference in my mind is the difference in the way the cells generate energy. And all of the cells in our body need to generate energy. And otherwise they die. You know, I mean, energy is energy. So our whole bodies, we breathe in oxygen and we give off CO2 and produce water as byproducts of aerobic respiration.
So aerobic respiration basically is how normal cells or healthy cells make energy. They take the protein, fat, and carbs with oxygen, that's aerobic, equals energy.
The tumor cell, on the other hand, pretty much functions without oxygen. So in order to function without oxygen, this is referred to as fermentation.
So the term fermentation is a primitive or ancient system of how cells can make energy without oxygen. The two main fuel sources for cancer are sugar and glutamine, which is a protein.
So all tumor cells, as far as I can see and as far as I've looked at, are more dependent on fermentation for energy than they are for respiration for energy, using oxygen. So this is why they live in hypoxic environments. They can survive in these environments because they primarily ferment. And when you ferment, you become limited in the types of fuels that you need, because oxidative phosphorylation, respiration, is an extremely efficient way to generate ATP, energy.
So the term oxidative phosphorylation is the final stage in respiration, which is how normal cells or healthy cells make energy.
You need a little bit of material like glucose or some other fat or whatever, and you can generate large amounts of energy just from respiration. But if you don't respire, the fermentation pathways are less efficient, therefore the substrates have to be present in large amounts. So the tumor cell is dependent on large amounts of glucose and large amounts of glutamine. And those are the two primary fermentable fuels. So the differences between a normal cell and a cancer cell is primarily the way they generate energy. The tumor cell needs to ferment. The normal cells can respire.
Got it. And can you touch on the origin of cancer? Because I think mainstream research is really focused on this genetic piece, but they're not getting into the metabolic or the damage to the mitochondria. Can you touch on that?
Well, I mean, that's the oncogenic paradox.
So oncogenic means something that causes tumors, and paradox in this context means something that sounds logical but leads to an unacceptable conclusion.
Cancer can be caused by many different insults to our body. Carcinogens, people look at that and they say, well, that's why we call something a carcinogen, because it has the potential to cause cancer in laboratory animals and humans. Radiation causes cancer. Viruses cause cancer. So the question is, how are these provocative agents causing cancer? The gene theory says that the carcinogen will cause mutations, and the mutations then lead to dysregulated cell growth. So you have to say, what is the primary characteristic of tumor? And it is dysregulated cell growth. So how does a cell under normal conditions become dysregulated in its growth? So, for example, a carcinogen is thought to cause a type of mutation which will then cause the tumor cell to become growth unrestricted. And if you look at radiation, is thought to cause mutations. Some viruses are thought to cause mutations, and these mutations were thought to be the cause of the cancer. However, every one of those same provocative agents also damages the mitochondria in the cell, the very organelle needed for normal respiration. Now, what happens is a carcinogen will enter into the mitochondria, leading to the formation of ROS, reactive oxygen species, which are carcinogens and mutagens.
So the term reactive oxygen species, ROS, is a reactive compound containing oxygen. Can be formed from within the cell as a byproduct of something, or externally from things like smoking, chemicals, drugs, radiation.
So what happens, the mitochondria produces ROS, which then causes the mutations. So the mutations are an effect of the damage to the respiration, not the cause of the cancer. The cancer and the mutations are both effects of the damage to the respiration. So this changes the whole game as to what the nature of this disease is. And we went through and documented very carefully all of the different agents that cause cancer, and every one of them first damages the respiration, leading to ROS, leading to the mutations. So this whole concept of mutations is mostly nonsense. It's absolutely a misdirected effort, which is and the tragic consequences. Over 1,600 people a day are dying from cancer as the result of this tragic misunderstanding of the nature of the disease.
Wow. So have you ever done any research or heard about research being done using something like IP6 phytic acid as a powerful antioxidant to counter some of the free radical damage?
Well, that's more, it would be in line of prevention. Mm-hmm. So, if you can prevent free radical damage to your mitochondria, you're going to prevent cancer.
So a free radical is an atom with an unpaired electron. Normally atoms have paired electrons. Free radical has unpaired, so it makes it highly reactive. Antioxidants give up their electron to stabilize the atom, making it neutral.
Cancer does not start with normal mitochondria. So things that would be reducing reactive oxygen species or inflammation or things like this would all be important in preventing the disease. But once the disease starts, you have an altogether different strategy that's needed.
Wow. Are there microscopic cancer cells in pretty much everyone? Is there any data on that?
I'm not sure about that. I mean, most healthy tissues, you don't see cancer cells. You only see cancer cells when they become enough to see. They make these statements. We all have cancer.
Yeah.
We all, those cells are usually destroyed. And taken care of. Well, we turn over our cells all the time. When a cell gets damaged, it's turned over. I mean, we have a maintenance system. They would turn over the cells. A cancer cell, it would be a cell that would not be turned over. Now, do we have precancerous lesions? I think if we have focal inflammatory sections, we would have certain, these are referred to oftentimes as polyps or something along these lines, small growths and things, but they're generally benign. They're not of a more invasive malignant characteristic. So I guess you can say that we probably have these things, but it's only when the tumor cell begins to invade the local environment and proliferate that we have an issue. Most of the cancer research today, in contrast to what we've defined, is focused on the gene mutations associated with the disease. So the concept of personalized medicine in cancer involves the development of drugs that will target specific mutations that are found in various tumors with the hope of correcting the disease. The problem with that strategy is that there are literally thousands, and some people have even estimated millions of mutations in tumor cells, which makes the targeting of any one or a group of mutations a pretty daunting task. And most of these so-called targeted therapies come with a significant amount of adverse toxic effects, which I've always found strange, that if a personalized medicine is supposed to target and kill cancer cells specifically, why do the patients become so violently ill as the result of this so-called personalized medicine? It appears to me that many of these drugs have a very significant off-target effect on the body as well. Our approach to this disease is basically a non-toxic metabolic approach that targets the metabolic deficiency of the tumor while enhancing the health and vitality of normal cells. A very striking contrast. It's a disease of mitochondrial function, which is the key metabolic component within the cell. And when mitochondria become dysfunctional, one of the possible consequences is the origin of cancer. Otto Warburg found many years ago that the cancer cells are primarily fermenters of glucose. They take glucose in, they metabolize it up to the point of a molecule called pyruvate, which is then fermented. And it seems as though almost all cancer cells will have this property. And the reason for this is because their mitochondria have suffered some sort of insufficiency or damage. And as the result, the tumor cells then must ferment, which is a very primitive form of energy, in order for them to survive. So most tumor cells will either ferment glucose or possibly amino acids like glutamine for their survival. So this then allows these cells to grow and persist. You can prevent cancer simply by keeping your mitochondria healthy. And what I mean by that is that with age, your mitochondria get inefficient. And this is a natural aging process. That's why cancer is more common in older people than younger people. Therapeutic fasting is a great way to enhance the functionality of the mitochondria to prevent cancer. You will not get cancer if mitochondria are healthy. It's just mitochondria are damaged by a variety of different things in the environment: chemicals, radiation, inflammation, viruses. Even inherited gene mutations that damage certain kinds of these mutations will damage the functionality of the mitochondria, thereby leading to inherited forms of cancer. But by and large, we can prevent cancer if we can keep the mitochondria healthy.